right, welcome to V-VSS. In this talk, I'll be giving an overview of an ambitious project we've been working on for a while now called the Natural Scenes Dataset, or NSD. Data collection involves scanning several human participants every week for about a year while they viewed thousands of natural scenes. Data collection is now complete, and we're excited to share it with you. Typically, in visual neuroscience, the data sets are relatively small and aimed at tackling very specific questions. The limitation is that these data sets may not generalize very well. We felt there was a need for something much bigger and much broader. More subjects, more sessions, more stimuli, more trials, and higher resolution. There's growing concern about reliability of fMRI results, but with such a large amount of high-quality data per subject, we can dispel some of these issues. The goal of this project was to collect a benchmark dataset that would be suitable for many different types of analyses and would also be enticing in the context of machine learning. So it needed to be a broad-reaching, massive amount of high-resolution whole brain data, a huge number of trials, and a huge number of visually rich stimuli that could help answer a variety of scientific questions about vision. Additionally, we wanted to focus on improving data processing methods. We had several priorities for this dataset. First, we knew it had to be big, meaning a lot of data from each subject and a sizable amount of subjects. It was important to us that the data have high SNR and high resolution, which is why we went with 7T fMRI. We also had a rigorous screening process when selecting our subjects. Third, it was important for us to be at the cutting edge in terms of our acquisition methods and analyses. Lastly, we needed to be hypervigilant and meticulous in terms of keeping track of study details and having very thorough documentation. Here's a general overview of the study. We had 8 fantastic subjects that came back roughly once a week over the course of a year. In those sessions, they were each exposed to between 22,500 and 30,000 trials of natural scenes. We acquired whole brain data on them, including cerebellum and subcortical regions. And this was done at a fairly high resolution of 1.8 millimeters isotropic. During this time, the subjects were participating in a long-term continuous recognition task. The images fit in a square of 8.4 by 8.4 degrees visual angle. The size was chosen to ensure participants could see the entire image with both eyes through the bore of the scanner. Images were presented on a Cambridge Research System bold screen. The images we chose were from the publicly available COCO dataset, which stands for Common Objects in Context. A nice thing about this dataset is how rich the images are, spanning many categories, and the images come with a large number of annotations as well as object segmentations. You can learn more about their dataset from their website, cocodataset.org. The experiment was a rapid event-related design in which each image was presented for 3 seconds, followed by a blank screen for 1 second. The subject's task was, for every image presented on the screen, to indicate if it was new to them or one they had seen before, either during that particular session or during any other session. So it was a challenging task. Here are a few trials to give you a sense of the pace. Note that they were instructed to fixate the central dot the whole time. Here's an overview of the data that was acquired. The dataset has a whole slew of functional data. We have what we'll refer to as the core NSD dataset, which was the viewing of natural scenes. This is whole brain EPI data. We also have resting state data, retinotopy and category localizers, and more. We also acquired several field maps per session for distortion correction. We acquired a variety of anatomical images, T1s, T2s, diffusion data, angiograms, venograms, and of course, behavioral data from the tasks. Some physiological data, pulse and respiration, and even some eye tracking, including videos of the eye tracking sessions. And lastly, detailed information about every single scan session, including a screenshot of the slice placement, survey questions for the subjects about mood, stress, hunger, caffeine intake, how well they slept, their comfort level, the day and time of the scan, equipment used, general notes about the session, and so on. As the data were being collected, we were assessing data quality, both in real time as subjects were being scanned, as well as through daily processing of the data as it came in. One of the things we came up with was a subject leaderboard to both monitor and motivate subjects on a variety of metrics, including their overall BOLD response, their response rate on the behavioral task, performance on the behavioral task, their motion throughout the session, as well as their within motion, or jitter as we called it, within run. Each subject is a different color. I won't go into the details of these here, other than to say they were all performing extremely well, and the blips you see are actually relatively tiny deviations. For example, the worst response rate was one subject on one day only responding around 96% of the time. Performance was generally at ceiling for every session. And motion jitter is on the order of sub-millimeter displacements. Here's alignment of the structural and functional data for all 8 subjects. That's the T1s, T2s, and EPIs. Here we're showing 3 views of each subject's brain, axial, coronal, and sagittal. What you can see is that alignment is very good for all subjects and the data quality is high. In addition to the great efforts put into pushing the boundaries with data collection, another important component of this project was data processing. Since we only had 3 trials of any stimulus per subject, and these 3 trials often fell on different days, we developed aggressive methods to derive good, robust estimates of single trial responses. A new way of getting hemodynamic response estimates for each voxel was also developed, which involved deriving a library of HRF time courses and choosing the best estimate for each voxel. This approach markedly improved signal quality compared to using a canonical HRF. We also use GLM denoise, which is a robust, data-driven denoising technique. Lastly, a new approach to applying ridge regression to the single trial estimates was developed. For detailed information, I encourage you to check out these recent related papers. We also have all of these processing techniques well documented and are happy to share details with you if you want to learn more. To give you a sense of how massive this data set is, here are single trial responses for one subject across 1,000 voxels for 750 trials by 6 sessions, totaling 4,500 data points per voxel. So across just 1,000 brain voxels for one subject, we're already looking at 4,500,000 data points. Now let's take a small square of this carpet plot and zoom in. Now you can see the individual data points more clearly. As you can see, it is a massive amount of data, which means more statistical power. Here's a video of BOLD responses for each subject across sessions, mapped onto flattened cortical surfaces. Various anatomical landmarks are labeled for your reference. Each frame is one session, and we're cycling through every single session, as you can see by the little counter in the middle. The big jumps you see are the shifts from one subject to the next. You can see how stable the BOLD response is from one session to another, as well as consistency across subjects. The strongest responses are in the occipital lobe, thank goodness, since it's a visual task. Here's another video demonstrating, on an FS average surface, brain regions generally responding to the NSD stimuli. These are group-level noise ceiling maps. Warmer colors indicate higher values, meaning more of the response variance can be attributed to the stimuli in those areas. Again, as expected, the highest noise ceilings are in the occipital regions. To get a sense of the behavioral performance on the long-term memory task, here are a few graphs showing adjusted hit rate, that is, hit rate minus false alarm rate, on the y-axis and time on the x-axis. The dots are performance pooled across all subjects, and the pink line is average performance, and the translucent pink is the standard error of the mean. The graph on the left delineates time between trials in a logarithmic spacing, going from repeats just one trial away out to 1 year away. The chart on the right shows the same data, but with time spaced linearly in terms of days. The horizontal dashed line is chance performance. The takeaway here is that performance is well above chance on this task, even when the image hasn't been seen for many months. Overall, this is very impressive performance. So not only is the brain data of high quality, the behavioral data is excellent as well. So this is a very enticing data set for scene recognition and memory research. The data could be mined many different ways in terms of memory effects. And we're happy to announce that we're making this a publicly available data set so you can access the data and analyze it however you see fit. We're on track to having a paper published about this data set and a public release of the data in early 2021. And you can check out our website, naturalscenesdataset.org, for more details and updates. If you're wondering what a dataset like NSD could be useful for, here are a few ideas, including exploring low-level features like edges, orientation, and color, training sophisticated neural networks, exploring individual differences, investigating cortical and subcortical regions, many possibilities. And if you want to hear about some analyses that have already been done with this dataset, I encourage you to check out Jan Charret's talk about semantic representations in the brain. The details are listed here. In summary, NSD is a large, high-quality 7T dataset on the perception and memory of natural scenes, designed to be broad enough to cater to an array of uses. The dataset will be freely available soon, and you can go to the link on the bottom for updates. And with that, I'd like to thank my incredible co-authors, our funding and resources, especially NSF and the CMRR for making the project possible, and thank you for your time.